Okay, hello students. We're going to finish up biology module 12 today, finishing up our study of plants. Is this the end of plants altogether? Let's see, 12. Yeah. Yeah, so this is going to be it for plants. And honestly, I really love this lecture a lot. I've always liked plants. I could have been a botanist, I think. But here we go. We're going to continue with plant structure and function continued, all right? So I mentioned a couple times last time about leaf color, okay? That all plants are green, right? Okay, so leaf color is due to the pigments found in the leaves. The pigments are found in different plastids of the cells. So the cells have these little organelles in them, right? We know that eukaryotic cells have organelles. And in plants, we have these special types of organelles called plastids, and these plastids can contain colors. One of the plastids you know about, it's called chlorophyll, right? Chlorophyll is inside the chloroplast, right? So plastid being the chloroplast, and chlorophyll is in there. And what color is chlorophyll? It is green. All right, so that's why plants are green. But we're looking at other colors as well. Okay, now this is a fun little lab you can do at home. Generally, what I would use would be a concentrated amount of some, plant chlorophyll, and then put it on a little piece of filter paper. But you probably don't have that. So what you can do is to take a leaf. One of the good ones to do with this would be a spinach leaf that you got from the grocery store that you're going to eat, right? And then just kind of rub it onto a piece of filter paper, namely something your parents might use in a coffee machine, like a coffee filter. A paper towel may work, especially the kind of paper towels that don't have all those little bumps all over them, like the Viva style. It might work, but then you might want to just try any other kind of paper that is kind of flat and not bumpy. Okay, and then you would just take the leaf, and what I did with my students, because it was easier, I just wrapped the leaf with a quarter, a coin, a quarter, and you can draw on the piece of filter paper with that, and because the quarter has that rough edge on it, it'll leave the green on the paper. All right, you can actually take a leaf and just draw with it on a piece of regular old paper, okay? And so then if you have some sort of filter paper you can put it on, then you can dip that filter paper just below the surface. You see how the chlorophyll goes here. So I rub my leaf right here, and then I dip the tip of the paper. The reason why filter paper works is because the filter paper is very absorbent. So paper towels are as well, so they might work. And you put a solvent down here, and what you can use is either alcohol, rubbing alcohol, or acetone. Acetone is fingernail polish remover. Okay, so you might know somebody in the house that has something like that. And you'll put that here, and then you'll watch the green actually travel up. And with with spinach, you'll see a green, and you'll definitely see like a yellow color on that. So that's a fun little lab you can just do at home. All right, now, I always have students just complaining to me about not all plants are green, and I always said, yes, all plants are green, but I found one. This is a non-green plant. It's called the Indian pipe plant. It is a saprophytic, so we know what that means, right? It eats dead things like a fungus, right? But it's an angiosperm. Remember what that means? That means it makes a flower. So here is this plant that grows similarly in similar regions as a mushroom would grow, as a fungus would grow, but it ends up making a flower. Look at that. Remember, what's the job of the flower? To make seeds, right? So it's sort of a bridge between the fungal world and the plant world, okay? But it is not performing photosynthesis. It just simply is not. So I guess some botanists would put it in the kingdom Plantae because it makes a flower, but other botanists would probably not include it because it doesn't perform photosynthesis. But that's the only one I could find, and I will have a little lab toward the end of this to show you how we can illustrate how indeed all leaves are green. All right, so chlorophyll. So we know chlorophyll is green. All right, and the reason it's green, it's all about chemical reaction going on, okay? So chlorophyll is green because there's an interaction between nitrogen, right here, nitrogen atoms, and a magnesium in the middle. So the way they're hooked up to each other like this, and we're going to see there's a bunch of carbon rings and hydrogen and oxygen, that kind of thing over here. And that is going to give off this green color for some reason. God picked green. He could have picked anything, but he picked green, and that's because of this large chemical, right? Now, here's an interesting fact that's not in your book. I just found it fascinating. Is that the structure of chlorophyll here with our magnesium and our nitrogens in the middle right here. Notice all these look, C20, those are 20 carbons coming off of there, all hooked on with some hydrogens as well. Every one of these little points right here represents a carbon. So it's very complex is my point right there, okay. Now, but this right here, notice, is similar looking. It's different down here. And then look in the middle. That's iron, and the iron interacting with the nitrogen, this is hemoglobin. And when the iron interacts, it makes a red color. I find that fascinating. What is that? So chlorophyll is the functional unit of energy in the plant, and hemoglobin is the functional unit of the blood system of mammals, right? It's so cool. And also I find it interesting, it's red and green. Yeah, did you know those colors are opposite each other? I don't know, I just find God to be so amazingly creative, yet he uses the same kind of, structure for different things. Very, very cool. Just an interesting little bit of trivia there. All right, so back to our plants and the plastids. Remember, plastids are specialized organelles in plants. There are other colors there that result from different pigments, sorry. So our chloroplast has green pigment. Then we have these things called chromoplasts. We're going to see some reds and oranges and stuff like that. These up here are kind of our stem cells up here. Our proto and an EDO right up here. Leuco would mean white. We have some white here. And then we have our amyloplast, elioplast, and proteinoplast. Okay, so proteinoplast would be where the proteins are. But we're going to see there's going to be other colors available. And when we mix some of those colors together, we get some pretty cool stuff. So most leaves are green due to chlorophyll, but the other pigments, so most leaves are green, all right? I'm going to tell you all leaves are green, but most leaves look green. But some of them look a different color because they might have these other pigments. The other pigments are called carotenoids. Those are the oranges. Anthocyanins, those are the purples. And the phycobilins, those are the blues. Okay, interestingly, if you find these beautiful colors naturally in your food sources, those are fabulous nutritionally for you. So good for you. So if you think of something purple that you can eat, like a blueberry. A phycobilin is probably blueberry. That's blue. Those are good for you. Carotenoids, you see the word carrot in there, right? So carrot, they're orange. Also, pumpkin and yellow and orange bell peppers, red bell peppers, those are all carotenoids. All right, so this is that other experiment I was telling you about. Okay, we're going to be looking at the anthocyanins. It turns out we use the anthocyanins that we find naturally in this particular plant called purple cabbage, and we use it for chemistry. It's very cool. So you can get purple cabbage at the grocery store. I love purple cabbage. You may not like it. If you've ever had coleslaw, you probably had purple cabbage in there, and you can eat it raw that way. Sometimes in your salad, you'll find some, sprigs, if you will, of purple cabbage in there. It's purple naturally, therefore, guess what? Very, very healthy for you, very good for you. But we have this really cool thing you can do. So here's what I'm going to tell you to do. Get your folks or somebody to buy you a head of purple cabbage and take about a quarter of it or so, and you're going to, I guess you have a lab in your book. Let's see, I have your book open here, and I'll see if I can find the page number. But what you're going to do is you're going to cut up the purple cabbage, and you're going to put it in a pot, and you're going to boil it, all right, on the stovetop, carefully, of course, maybe with parental, help. And when you boil it, the boiled water will turn purple. So then you throw out the cabbage. You can eat it if you want. It's delicious. Boiled cabbage with some butter and some salt. Yum. Use the rest of the cabbage, by the way, the stuff you didn't boil, to make some nice, yummy coleslaw. Good recipes available online. Anyhow, so you're going to take that liquid, let it cool off a little bit, and pour it into a clear glass. It'll look purple. And then, maybe a small glass, add some drops of vinegar to it. And you're going to see something really cool. It's going to change colors. And that's because it turns out that purple cabbage, the anthocyanin that's in purple cabbage functions as what we call an indicator. So an indicator solution, okay? And when the pH changes, vinegar is very acidic, so it's going to change. I did find it in your book. It's on page 497, and it'll tell you exactly the step-by-step procedure to go through and to look at it. It says to use vinegar, but it also says to use ammonia. Ammonia is a natural base. You probably have it. That's the opposite of an acid. You probably have some in your under your kitchen sink. You might use it for washing, for mopping floors, that kind of thing. All right, so give it a go. It's an easy experiment. It's totally fun, and then you'll have purple cabbage to eat too. All right, the word deciduous. Deciduous means to fall off or fall out, okay? A deciduous plant is a plant that loses its leaves in the winter. Look outside and you'll see a lot of trees out there with no leaves on them. Those are deciduous trees. Interestingly, baby teeth are called deciduous because guess what? Your baby teeth fall out and you get new teeth, all right? Okay, so that just means it falls out or falls off. So a lot of plants are deciduous. They do lose their leaves. Not all of them do. Look out. If you see something green right now in the wintertime, that plant kept its leaves. Usually they're sort of in the pine or the spruce family, right? So we call those evergreens, right? They keep their leaves during the wintertime. Okay, so here it's interesting. Why do plants lose their leaves? So generally the reason why the plants go into a dormant time during the winter is really to conserve their energy, right? And so they have shorter days, therefore less sunlight, therefore not as much opportunity for photosynthesis to take place. And so what they'll do is they'll drop their leaves off and not have to do any work. They'll take the winter off, if you will. Okay. So that's the reason they do it. But now we're going to see the way they do it. Okay, the mechanism on how it happens. All right. So the xylem and phloem are blocked by the xylem and phloem headed to the leaf is blocked by something called the abscission layer. Okay, as the days get shorter, it notices how much sun it's not getting. And what will happen is these chemicals, we're going to look at these chemicals, the names of these ethylene and oxen very quickly here in a few minutes. It shuts off the water that goes to the leaf. And if the leaf doesn't have any water, we know what happens, right? It's going to turn a color. The green's going to stop doing photosynthesis, therefore it's not green anymore. And then we're going to see those secondary colors appear. Now down in Texas, typically our secondary color is brown. I call that tannic acid. But some trees will turn yellow or red or something like that. Those of you who are, blessed to live in places like Virginia or up on the eastern area there, you can see what we call, the changing of the leaves, right? The foliage changes in the fall. And that's because the green is gone and the colors remaining will be visible, usually yellows and reds. All right, here's a close-up look at it, the abscission. The leaf abscission right here. So what happens is those chemicals come in and break that so that water can no longer pass between here and here, okay? And then it'll drop right off. And if you take a look at some plants right now, you can see on the branch, there'll be a little place where a leaf used to be. We call that a leaf scar. A leaf scar right there. All right, so pretty soon though in the spring, you're gonna start seeing new leaves coming out. Here they are just falling. Look at those beautiful colors. We don't have that in Texas so much. Mostly brown. Yeah. Brown is good too. That's how white tea is brown because the leaves were brown. Yeah. Black tea, they call it. Okay, let's look at water and nutrient transport. We know that water and nutrient transport is taking place in the vascular tissue, right? Remember what those are called? Xylem and phloem. All right, there are 4 processes that those are going to use. Here we go. We have photosynthesis, turgor pressure or turgor pressure, hydrolysis, and transport. All of these are how we are getting nutrients and moving nutrients, okay? All right, so turgor pressure right here in plants, this kind of reminds me of our guard cells last time we talked about, how they would get real plump and then kind of spring open, and then if they didn't have as much water, they would get flaccid and kind of flat. So we're going to see here when they have plenty of water, this big central vacuole will fill up. Remember this from cells. We learned about cells. It'll fill up and make everything nice and tight in there. Where if we have less, it'll be like this. It'll be a plant that is wilting. In fact, I think I have a picture of that. Yeah, see, so when they're full, nice and full, this is good. If you haven't watered it, it'll go, right? Just think about a piece of celery. If you leave a piece of celery out accidentally overnight or whether the next day it's all bendy, and who wants to eat that? Nobody. But you make a fresh cut. You don't even necessarily have to make a fresh cut, but if you do make a fresh cut, put it in a cup of water. Guess what? Boom, boom. It'll be full and delicious in no time. All right, you can see the picture on the right here is a flower opening. That's a blossoming flower. It's opening and blooming. Okay, the process responsible for that kind of movement is called nestic movement, and it has to do with the turgor pressure. All right, it's responsible for some of the motion that we see in plants. So the change can be triggered by a change in the amount of light that's received outside, the length of the days. Some flowers will open their petals during the day and close them at night. We call that nastic movement. Have you ever seen a morning glory? Here, spring is coming, and you're going to see, I heard there was an early bloom of bluebonnets on the way. So have your eyes peeled for that. But morning glories, they'll open in the morning and they'll close at night. And then there's the evening primrose that opens in the evening and closes in the morning. Yeah, so that's it. So this is the official definition in your book of what nastic movement is. Okay, and if you ask me, it's overly complicated. Nastic movement in plants is the response of a plant to a stimulus such that the direction of the response is pre-programmed and not dependent on the direction of the stimulus. And all that means is that it is going to make the movement it's going to make, okay? Depending on whether it's the light or whatever, the water, that kind of thing. So let me give you an example of that. I think that's just an overly complicated thing. So here we're going to look at, this is called sensitive plant. Notice it was touched, and now they're closing up, right? They're touch, and they're moving. Sometimes if you tickle it down the middle, it'll just close. This is called the mimosa plant. We have these around here in Texas. That's pretty cool. And so other plants you can see, this one is just happens, quickly right in front of your eyes. That's not even a delayed video. It's happening that quick. So that's kind of fun. So we call that the sensitive plant. All right, hydrolysis, when the plant cells take in larger molecules, bigger ones that have to come in there, they have to be broken down to be useful molecules. This happens by way of hydrolysis. What we do is we're going to add water to it. You see right down here, add water to it, and we're going to make it into something else that's easier for the plant to get a hold of. Okay, so that's just the way. Hydrolysis just means I'm going to break things down with water. Once again, water being an important process in plants. Here it is. We have this. We add water, and look, we broke it into two by taking that H2O and adding them to the air. Look what I ended up with in this particular case. I took maltose and broke it down to 2 glucoses. Hooray. Yeah, glucose is good. All right, water absorption in soil. I think I mentioned last time we were talking about dirt today a little bit. Soil particles play a role in nutrient absorption, okay? First of all, they're in the soil. Remember, the roots are holding them down there for absorption, and the roots are holding them down there for anchoring, right? But in the soil itself, there needs to be room in the soil for water to be there, okay? We call that pore space, P-O-R-E, pore space. It allows for soil to hold water and air. It's important for air to be down there as well. I don't know if you know this, but plants can drown, If you have so much water, you can overwater a plant. You might have done that before. Yeah, so it's good to have nice friable soil that has room for air. A lot of times people would think, if you till up the soil with a tiller, that's going to loosen it and aerate it and that kind of stuff, which is true. But recently we've discovered that when you do that, you also disrupt the entire microbiome of the soil. And so people are starting to rethink that a little bit. And instead of tilling up the soil, they're spreading a lot of stuff on top of it, like, compost and things like this, which will make for a nice, rich environment of bacteria living in there, of different fungi that are helpful, like, mushroom type things. That'll be helpful for the plant. And they are, that will make the soil friable on its own. Okay, it may take more time. You have to be more patient that way. Some people call it lasagna gardening, where you layer down things, and over time it's going to really make a nice, loose, friable soil. So let's take a closer look at that. So wherever you live, you might take a look at the naturally occurring soil that happens there. So in our area, Texas has all the kinds of soil. There are some places that are very sandy. We have a place out in our area called the Tahitian Village area. It's very sandy. Now, sand is nice for pores, in other words, lots of air in there, but it's terrible for holding on to water, right? Because think about if you have sand, the water just flows right through it. Now, where I live, I have a bunch of clay. Now, the thing that's great about clay, it's got a lot of nutrients in it. It holds water great, but it has no room for air. So plants can't grow as very, very well. I've never had very good luck growing even grass in my yard. We have silt. Silt is sort of like if you, I'm going to show you, it's just smaller. It's just a smaller bit of that. When we mix these up in a beautiful, recipe, we end up with what everybody wants in their garden. It's called loam. And generally we want something known as sandy loam. Okay, so loam with sand in it, giving us the nutrients, the ability to hold on to the water, and some pore space for the air. Okay, so the particle size of soil from the largest to the smallest. Okay, so gravel is the largest, then comes sand, then comes silt, then comes clay. So let's go back again to our previous slide. All right, so which is the biggest one? Gravel's not in there. Then comes sand, and then silt. And look, clay's the smallest. That might be hard to see in your mind's eye, so I'm going to show you a artist rendition of this. So if this is gravel, and gravel can be several different sizes, obviously. Then if you ever looked up sand really close, like with a magnifying glass, it's pretty cool. It's like little tiny, boulders in there, but they're itty bitty. Then comes silt, and see, clay you can't even see on this scale. But here, if my sand were this big, my silt would be this big, and my clay would be that big. That's how much smaller clay is. And that's part of the reason why it doesn't get good airflow, is because the pieces actually, they shouldn't draw it as a circle. They should draw it as a little flat thing. I think I might have a picture of that. So here, yeah, look at our beautiful loam. Every gardener just dreams of this kind of soil right here. Loam is a mixture of all the particles plus some organic matter. So what is organic matter? That would be compost, right? Old leaves from trees are perfect for that. If you have a compost pile, you might be taking the scraps from the kitchen, eggshells, things like that. And all of that works itself back into the soil. And it's really, really good for growing plants. Lots of good nutrients in them. So how to do a squeeze test. Here you go. We have the picture here. So if you take some soil from your garden, and it clumps immediately but falls apart when the soil, then it's too sandy. If it stays together and doesn't crumble at all, it's too much clay. But if it's silty, it'll feel slimy when wet and become powdery when dry, so that's kind of okay. It comes a little maybe a little too powdery. Ideal loam soils when you hold it shape, but then you poke it and it crumbles up. Okay, so try that with the soil at your place and see what kind you have. If you have too much sand, you might want to add some clay type soil that you can buy somewhere. If you have too much clay, you add sand. Yeah. All right, back to leaves. We're going to be looking at one of our next things as far as water, in plants. We have something called water transportation. We know this. And then we're going to be looking at transpiration. Oh, I think I said transportation before, didn't I? Okay, so transpiration makes more sense. Transpiration is happening in the cells. Now we can see, we should recognize this from last time. Here's our guard cells and our stoma, right? And remember I told you water's coming out of that? I showed you that little schematic of the water flowing out of that. Okay, so that is transpiration. Okay, notice it shows us the underside of the leaf right here is going to have this right here, and we're going to see how water transpires. Okay, we use capillary action to pull it. Water's coming from the soil, up through the roots, up through the stem, up to the leaves. As long as the leaves have plenty of water, it can release some of it through those open stoma. Remember, the stoma won't even be open if it doesn't have enough water, so it doesn't do that when it's all wilted. Okay, these are only very healthy, hydrated plants that do this. All right, so we can see it evaporates. Pretend like this picture is, we're looking at the underside of a leaf, because that makes more sense to do that. So then the water evaporates. That's why it's cooler to stand in the shade of a tree, like I said last time, rather standing under the shade of a building, a pole barn or something like that. All right, see, it's going up. Now what's causing it to go up? We call that capillary action. Okay, it's like when you take your straw and put it in the water, right? It'll go climb up the straw. So we're going to see here the reason why the capillary action takes place. That's water going through the xylem and the phloem. I think that's the phloem right there. Going out. Ooh, check it out. There's our little guard cells and our stoma. All right, so it's coming up. And it's going to flow through the leaf. Remember, the leaf needs the water for photosynthesis. It's going to flow through it. And if it has plenty, it's going to go right on out. Okay? So because this is going through this hole, it has this action, this movement, and that encourages more capillary action. Pretty beautiful. Yeah, so here we go. It's moving upward naturally against gravity, which is pretty cool because of the little straws, the little tiny straws. Remember, xylem and phloem in there. And it's going to come up, and because it's being lost out of the leaf by transpiration, going through the holes like that, that actually keeps up. I don't like to use the word suction at all. It's not really a real thing. It's really about the changing of pressures in various areas like this, but it sort of is So like even the word suck, it's not really, when you're sucking through a straw, for instance, it's not your sucking that's doing it. It's you emptied the air out of the straw by doing that. And because of that, the pressure on the top of the liquid pushes it up the straw. So it's really pushing it up versus pulling it up. I know it gets a little complicated. This is another experiment. Love it, love it. Just any house plant that you have, if you don't have a house plant, go buy one. Just because it's nice to have living things in your house, right? And in part of the plant, you take it and you put a bag around it and put it in the sunshine, inside if you need to, if it's cold out or whatever. And you're going to see over some time, guess what's going to happen? Oh, look at that. The water's going to, this really works, y'all. It really does happen. And so give it a try, and you're going to see the transpiration's going to let that water into that bag. Pretty, pretty cool stuff. All right, now let's talk about water a little bit, because water is indeed a miracle. Thank you, God, for water and for making it the way it is. Okay, so water is H2O, right? We can see our H2O, two hydrogens and an oxygen, and we're going to see these properties that it has, okay? So let's say we're inside a, tube of xylem inside of a plant. So what happens is that the water, it adheres to the side of the xylem, but it also holds on to its fellow water molecule. We call that cohesion. So adhesion and cohesion, they're holding on to each other. This, of course, is a simplified picture. There is actually a bond between them called a hydrogen bond that keeps them together, just because of the polarity of water, where we have sort of a partially negative charge on the hydrogen and a partially positive charge on the, hydrogen, partially negative charge on the oxygen, partially positive charge on the hydrogen, and that makes this hydrogen attractive to this oxygen, okay, because of the negative positive polarity situation. All right, so water has a cohesive characteristic, and cohesion tension theory is based on the pressure, notice there, pressure, not suction, in the xylem moving water up from the roots to the leaves. Okay, so because of that cohesion tension theory, and we call that capillary action, moving up to the leaves, away against gravity, right, which is difficult. So here's the water, the red and white things are the water, right, okay? And they're adhesing to this side, but they're cohesion to each other. That's a hydrogen bond. Notice the white is attracted to the red. That's that polarity situation, okay? And it has that cohesion tension thing. All right, translocation allows for transport of substances throughout the plant in the phloem. All right, so notice the xylem. Remember, it's dead, right? It's just a tube. Transpiration of the water going up this way. I mean, we could say transportation, too, in that case. But then we have the phloem, remember phloem down, but it is kind of flowing both ways, really, because we have these living cells here. But look, it says sucrose. It's taking the sugars, it really should say glucose, glucose down to be stored in the root, be stored in the stem, that kind of thing. So translocation. All right, so how do all these things happen? Well, there is a lot of work being done right now trying to learn about plant hormones. There's plenty of work left to be done here because there's still some mysteries, okay? But plant growth and responses are controlled by hormones. Those are chemical substances. They are chemicals that circulate throughout multicellular organisms, regulating cellular processes by interacting with specifically targeted cells. Okay, it's interesting because that's the same thing that happens in the human body. We have certain hormones that we send out and certain cells that receive it because they have a receptor on it. Those are called target cells. They respond to whatever the hormone is telling your body to do, okay? So the same thing is happening in plants. These chemicals are telling the plant to do something. So let's look at the hormones. We have these 6 that we're going to key in on, and you never know, there might be more. All right, here we go. Auxins, that's the one we've known about the longest, okay? We have cytokinins, we have gibberellins, ethylene, a gas, abscisic acid, which we talked about in the abscission layer there just a few minutes ago, and something that's the newest one we know about called Florigen, okay? That we didn't know about that for a long time, so we're still learning so much about these. All right, the auxin being the most famous one. What we think is happening here, remember, once again, we're back to science is very good guesses, right? So auxins affect plant stem growth through processes known as phototropism and gravitropism and thigmotropism. I'm going to go through each one of those. So, but what happens here is that when the sunlight's hitting a plant, you may have noticed this if you have a house plant, you might see that it's leaning toward a window, right? And if you turn it around, in a couple days, it'll be, leaning again. So in other words, it will lean back toward the sun. We believe that has to do with auxin effect, okay? So if the sunlight is way overhead like this, right, the auxin molecules will all be at the top, closest to the sun, and it'll go straight up. But if the sun's over here, the auxin molecules will move to one side, causing elongation on the one side, making the plant tip toward the sun. You've all seen probably pictures where they show maybe a time lapse of sunflowers following the sun. It's true, okay? So this process here is known as phototropism. Tropism to move, photo by way of light movement, okay? So if that's the case, then we have gravitropism. What do you think that is? Movement by way of gravity. And then thigmotropism, movement by way of, you might not know this word very well, but it means touch. Remember that sensitive plant that we saw, boom, the nastic movement, that's thigmotropism. All right, so here we go. Gravitropism, it knows which way is down. Thigmotropism, it knew something touched it, okay? And then, so gravitropism is going down. Here we go. Hydrotropism, oh, it's going toward the water. That's another one, hydrotropism. So that's one thing about seeds, they know which way is down. That's crazy, isn't it? They've done some interesting studies in the space station about what do seeds do when they germinate in space? Because there's no gravity, right? So no, thigmotropism is feeling that little rock and moving around it. So the cytokinins, they affect the mitosis rates, okay? So it will stimulate cell differentiation or inhibit it, okay? So if times are rough, it'll slow things down. If times are good, it'll speed things up, okay? So CKs would be our cytokinins right here, and they're going to cause branch, the shoots, the meristem differentiation, flowering, all that stuff, the sugars being made, and then sometimes it will slow things down, say in the wintertime, right? For trees, it would slow things way, way down. Gibberellins, the elongation of stems and leaves and fruit growth. We can see a picture here. These were treated. Somebody actually sprayed the plant with gibberellins. So that might be a little bit of a manipulation of plants. I don't know if you have a problem with that, right? GMOs, a genetically modified organism. I don't know if that wouldn't be genetic in this case. If you're spraying it, I think it goes on it rather than in it. A GMO would have something going in it, like changing its DNA kind of a thing, which I think could be a little scary. Yeah, we should go back to the way God planned it, in my opinion. But the gibberellin hormone does cause the fruit growth and flower growth to be more prolific. Ethylene will cause fruit to ripen. Now, this is interesting. You probably noticed that. The banana is a really good example here, that if you buy a banana in a store and you let it sit on your counter, it will ripen, right, as it goes. And that's because it has a certain gas that sort of goes around it. Did you know if you put it in a bag, it would ripen more quickly? You've probably heard that about avocados, right? You put them in a brown bag, and that's because you're keeping the gas around it, right? So that helps to do that. So if you buy your, let's say you buy your banana at stage 3 right here, and you get home, and really the best time to eat it probably here, right? Stage 5. If it gets here, you're ready for banana bread, I think. It gets a little too overripe, if you will. So that brings us to this idea of fruits that ripen after they're picked and some that don't, okay? It has to do with the ethylene action, and we call that climateric. So we have a climateric fruit and a non-climateric fruit. So our climateric fruits are the ones that will ripen when you bring them home. All right, some of these might surprise you. I was a little surprised about apples, honestly. I thought that they just stayed, because apples last a long time. We all know about avocados. You buy an avocado when it's hard, you set it on the counter and it softens up when it's time. Bananas, we already talked about that. Turns out plums, blackberries, tomatoes, you can take a green tomato from the garden and put it on your counter and it'll eventually turn red. But you might also notice that they're deeper red when you leave them on the vine. Okay, so on the vine they will turn a bright red, but you got to bring them in and eat them pretty quickly, right? So pears, I kind of like with apples. Remember, those are both plums, right? We talked about plums. Peaches, yes, those ripen more. Apricots in the same group, and kiwi. Non-climateric, in other words, these do not get any better, all right? So if you get oranges and bring them home, they don't get more ripe as you let them sit there. They just rot. Strawberries are a great example of that. You get your strawberries home. If you don't eat those in the first couple of days from getting them home, boy howdy, they're going to be moldy, right? They'll just go straight from good, beautiful red strawberry to moldy and kind of goopy, right? I will say if you rinse them with like a vinegar rinse, sit them in like a diluted vinegar water for a little while. It takes off any kind of fungi that might be on them, and they'll last longer. They won't mold as quickly. Lemons, I keep them in the fridge. Grapes, we just eat them within days when we get them. Peppers, we keep in the fridge. Remember cashews? We had that whole discussion. Not the nut, but the cashew apple, right? Once it's ripe, that's when they pick it so they can get the cashew off of it. But I told you we don't have the cashew apples here because they can't ship them to us because they've ripened already, and they just rot afterwards. Okay. All right, abscisic acid, it is the hormone that inhibits the abscission layer from forming to stop growth during stress or dormancy. This is the whole leaf, losing the leaves thing. So we discussed that. In the shoot, okay, we can have the abscisic acid in the shoot as well. Look, it causes the stoma to close in seed plants. The reduction of leaf mesophyll, you know all this stuff, right? In the root, it induces root elongation. It inhibits lateral root formation. And that would just kind of, during the winter, it'll slow down. During the spring and summer, it'll speed up. Now, Phlorogen is the hormone that we think controls flowering, right? We know nastic movement. We know that water has to do a lot with this. Transpiration has to do something with this. But we think there's also a hormone, and we've called it Phlorogen, meaning having to do with flowers. All right, here we go. Phototropism, responding to light. Now, look at this really cool experiment here. So we have the light over here. The uncovered one, this is our control. It's going to head toward the light. Now, they covered the base of this one to see, okay, well, look, it's going to the light. But when they covered the top of it, it did not bend toward the light. This is why we have the hypothesis that the auxins causing it to bend are only in the tip. Okay, so this is an experiment that's been done. That's pretty cool. All right, gravitropism, the plant always knows which way is down. So it sends its roots down every time, and it shoots up, right? So notice I lay it on its side, it's still going to go up. You've probably seen trees that are growing on the side of a hill or something, and the trunk still heads upward, right? Because it understands where gravity is. And that's because of a hormone. Here's stigmatropism. It touches something. Watch what it's going to do. Yep, it's going to grab that. So it felt that it had this is a plant that would be growing like a vine up something, right? So that just, this is sped up photography, right? It takes a while for that to happen. You know, it might happen overnight. You'll come back the next day, it'll be holding on to something. So that's stigmatropism, responding to touch. In fact, that sensitive plant, that's the same thing happening there. So that hormone and nastic movement. All right, we're going to look at a couple of unique plants. This one right here is called the corpse flower. There's a couple of different ones called corpse flower. So this one is one of them. It's quite large, actually. It's probably, I'm going to say, I don't know, 3 feet in the center there. It's big, you know, and it stinks. It stinks like rotting flesh. And that draws some creatures to it. You know, some creatures that are interested in eating something like that. And they'll end up falling inside. They're sort of like an insectivorous plant, that kind of thing. Some of them be drawn that way. There's another one, though, the Titum arum. I want to show this to you. I think I have a video of this one. This is the one that blooms the one time, but it's real stinky. Is this the video? No, I think maybe the next one. Let's watch a video.
Maybe only once a decade and lasts about 24 hours. The bloom of the rare and gigantic Titum arum, better known as the corpse flower. It's a mimic. It copies the stench of dead, rotting animals. But hold up. What you're looking at isn't actually the flower. That part, it's a modified leaf called the spathe. It even looks like raw meat. And this is the spadix. It heats up like a mammal's body. So warm it steams. It's all meant to attract its pollinators, insects that lay their eggs on fresh carcasses. But the flowers? They're way down in here. Each of these is a male flower. and below are the female ones. With just hours to reproduce, the stakes are high for Titan arum. It would be easy if it could reproduce using its own pollen. But the plant needs fresh genetic material, pollen from other corpse flowers, to make these, the fruit and seeds that eventually will become healthy new plants. So, it staggers things. The female flowers are ready first. They get sticky. The plant sends out its powerful stench, more than 30 chemicals in all. Some are nice. The spathe releases a jasmine smell. But mostly, it smells like funky cheese, rotting garlic, or dead rat in the wall. That aroma tricks flies to come investigate, thinking it might be a good spot for their young. And they might bring along pollen from another corpse flower, brushing it on the female flowers. A few hours later, the male flowers release strings of pollen. Some of it falls on the female flowers, but by then they're no longer able to use it, which is how the plant avoids pollinating itself. Titan arum is rare. Poachers and deforestation in Sumatra, where it's from, have taken a toll. And in botanical gardens, like here at the University of California in Berkeley, there aren't any other Titan arums in bloom to share pollen with. So when one opens up, biologists often collect pollen by hand to freeze and use later when another corpse flower is ready. Which is how this lonely giant survives, with such a foul and fleeting bloom.
Okay, well, that doesn't seem very advantageous in a ecological sense or whatever, so for evolutionary process there, the fact that the plant needs human intervention or whatever, that's crazy. So how it survived all this time, I have no idea. So anyway, some of the other ones, the other interesting plants that we're looking at are, we look at freshwater plants like water lilies, right? They actually grow in the water. I think I mentioned last time that plants don't technically need soil. They can grow in water, and so these water lilies are doing that. They're getting photosynthesis. They're probably getting nutrient from the water as well. There's probably, I think that ponds that support water lilies often also support other life, fish and turtles and things like that help to keep the water nutrified. Saltwater plants, these are more interesting because salt typically isn't great for a plant. If you spread salt in your garden, you will kill your garden. Salt's not good for it. It usually causes them to be dehydrated, pulls the water out of them. But these are specially designed to live in the salt water. They're called mangrove or mangrove forests. They actually do have salt glands on them. If you take marine biology sometime, you'll learn about these guys. But what's so cool about the mangroves is they have this dense kind of root system under the water, and it makes for like a protective region for animals to live, sort of like a coral reef. But here using the roots of the plant instead. But it is an actual plant, whereas other green things in the ocean tend to not be plants, and blue-green algae, things like that. Desert plants, here we go. We have our cactus. We talked last time about the green part of the cactus is the stem, and the needle is the leaf. And then, of course, these fancy ones, the ones that are only found, in my understanding, only found in Arizona, right, are the saguaro cactus. Here's a kind of the fancy ones here. I think these will grow into that, but with the fancy little parts coming on there. Saguaro is very unique to America. Here we can see, look at that. They're making flowers, right? And the flowers are going to make seeds. And then we know about insectivorous plants. Now, I think I mentioned that the Titan arum, it's not insectivorous, it just attracts insects for pollinators. Okay, so if I misspoke, I apologize for that. I think I have a really Fun video here. It's a little frightening. Some of the other teachers were kind of horrified by this one. Let's see if you like it. It's creepy.
The Venus flytrap.
Attenborough.
Like the sundew, it makes itself very attractive, oozing nectar across the brim of each leaf. But any visiting insect had better watch out for these 6 tiny hairs. This fly has to tread carefully. If it strikes one hair, it can carry on feeding, but a timer has been set. A second strike in less than 20 seconds, and the fly is doomed. An electrical impulse is triggered, and the leaf snaps shut in just a fraction of a second. The tips lock together like prison bars. If the fly is very big or very small, it may just manage to escape. But most are trapped and die.
Okay. I don't know if that creeped you out at all. It sounded like the little critter was, screaming there. That, by the way, was narrated by a very famous, guy called Sir David Attenborough. He makes excellent documentaries. I think he's like almost 100 years old, and he's still alive, as far as I know. All right, so if you get your purple cabbage, and you can take it and add different amounts of, like it says in the book right here, we can see, adding vinegar, and then, so acid would turn it pink. 7 is neutral right here. That's what color it comes out after you boil it. And then if you put, if you don't have any ammonia, another base that you should have at home would be baking soda. So try that. The problem with baking soda is that it might take a lot, and it kind of makes things murky. Because the baking soda sort of suspends in there. Anyway, it's a great lab, so it's in your book all written up, so I would recommend doing it. And then we will be now done with plants, and we're going to move into animals. Hooray! All right, everybody, I hope you have a great time studying with the plants, and I will see you next time.